Thank you very much, Christine, for the very kind introduction. You are very, very good TA. So, and thank you for the invitation and the opportunity to speak here. So today I'm going to tell you about how we can use the technology that was originally developed in physics and astronomy for high resolution, both in the spatial and temporal domain imaging of the brain. So the brain itself is one of the most essential organs in our body. It is made of neurons that communicate through very small structures called synapses. In the human brain, there are about 100 billion neurons that are making about 1 quadrillion connections among themselves. Even for the much smaller brain that we use to study neuronal computation in the lab, such as that of the mouse, there are about 100 million neurons making about 100 billion connections. Now, the ideal mind reading machine should have submicron spatial resolution, so you can see the biological processes that is happening in the individual synapses. It should have millisecond scale time resolution because this is how fast information transfer from one neuron to another. You need to be able to work in brain tissues that are not optically transparent over large volumes because that's how much the neural circuit extends itself. If you can express fluorescence protein in those neurons, then we can use optical microscopy for such a mind reading machine. One of the most popular methods for in vivo imaging of the brain is two-photon fluorescence microscopy. Here, a single fluorescent molecule will absorb 2 photons simultaneously in order to go to the excited electronic state. From there, when the molecule returns to the ground electronic state, the actual energy can be released in the form of a fluorescence photon. Because this 2-photon absorption process has a very small probability to happen, it only occurs at the focus of a high numeric aperture objective, where the light intensity is high enough. In order to generate an image, what we do is to scan this focal point in 3 dimensions inside the sample and record the fluorescence brightness at each position. And this gives us a micrograph of the fluorescent molecule distribution inside the sample. As you can see, the resolution of this method is very high, which allow us to see individual synapses as shown here. What makes this method particularly important for neurobiology are the development of genetically encoded activity indicators. Those are the fluorescent protein molecules whose brightness reflect of the neuronal activity. For example, here, each individual neuron are expressing a calcium indicator. Whenever the neuron becomes active, calcium concentration goes up, and you will detect an increase of fluorescent brightness around this cell. Because of the very high spatial resolution of 2-photon fluorescence microscopy, you can see this process even in individual synapses, as shown here. Now, ideally, we would like to be able to do such measurement everywhere inside the brain. However, several realistic issues prevent us from doing so. Therefore, one of the major efforts in our lab is to develop optical method so that we can image at higher resolution, larger depths, and much faster spatial scale. So the first approach that I would like to explain to you is that the problem of aberration. Ideally, when we want to generate 2-photon fluorescence, we want to be able to focus all the excitation light energy into a focus whose size and intensity is limited by diffraction. However, in biological sample, because of the refractive index inhomogeneity, the light rays do not intersect at the same point. They also don't have the right phase relationship. In other words, the wavefront of the excitation light is distorted from its ideal form. As a result, instead of a very tight and bright focus, now we will have an excitation focus with reduced focal intensity and enlarged focal volume. And this is really a problem for most of the biological samples, especially in vivo, because it's really just impossible to achieve this kind of diffraction limited performance, limited by the sample themselves. So this is not a problem that only microscopists encounters. For many years, astronomers know that if they want to use a ground-based telescope to look at a distant star, ideally they would like to have the starlight to form those diffraction limited spot on the camera. However, the atmospheric distortion of the wavefront of the starlight causes this very fuzzy images of the star. Now, if you are looking at the star in the center of our galaxy, they look all very fuzzy and you won't be able to resolve them individually. Back in the 1950s, it was proposed that if we can measure how this wavefront is distorted, then you can correct it and then recover a diffraction limited image of the star. It was only in the 1990s where the technology advances sufficient to implement this approach called adaptive optics. But once you can do that, you can see that for the same star, once you turn on adaptive optics, you will have a much brighter and refined image. As a result, now you are actually able to resolve the stars that are orbiting the supermassive black hole in the center of our galaxy. And this is really the technology that enabled Reinhard Genzel and Andrea Ghez to study the supermassive black hole, which won them the Nobel Prize in Physics last year. Now, we cannot actually directly apply the adaptive optical method developed in astronomy for microscopy because the sample we are looking at are optically opaque, unlike in astronomy case where they are looking through clear sky. So in my lab, we've been developing adaptive optical method for microscopy over the past 10 years, and they are all based on a very simple physics picture. In that, since the problem with sample-induced aberration is that the light rays are not intersecting at the same point, what we first want to do is to measure how far away each individual light rays are from the desired focal position. Once we know that, we can add additional tip and tilt to those light rays so that after passing through the sample, they will intersect at the same point. And then we just need to make sure all the light rays are in phase so that they can constructively interfere to give you a diffraction limited focus. So here I'm just going to show you a few example applications. So in this example, we combine adaptive optics with two-photon fluorescence microscopy, which allow us to look at those synaptic structures, those little protrusions from those fine processes at beyond 600 micron inside a live mouse brain. And if you compare the images that you would get without adaptive optics, you can see that the signal enhancement as well as the resolution improvement is very much essential in order for us to see those individual dendritic spines. Those are the part of the synapses that the neuron is using for communicating information. More recently, in the effort of improving the in vivo imaging depths, Chris Shi's lab developed a three-photon fluorescence microscopy where longer wavelengths of the excitation light can be employed to reduce tissue aberration. We combined adaptive optics with three-photon fluorescence microscopy, and in this example, we were able to image a neuron at 757 micron below the surface of the brain at a very high resolution with the help of adaptive optics. If we compare the brightness of the cell body, AO increased by about 7 times. And if you compare the brightness of a dendrite, the brightness increases about 19 times. We can zoom in further, and you can see that again, it's really essential for adaptive optics in order to observe those synapses. So here, this dendrite signal is improved 16 times. And we can zoom in even further. You can see that only after AO, we can see dendritic spines and even dendritic spine neck. So this is how we can improve the spatial resolution of in vivo imaging. But how about the temporal resolution? As I mentioned to you previously, in order to form an image, we have to scan this focus around inside the sample. If you scan a focus in 2 dimensions, you will get a cross-sectional view of the structure, which is not enough to tell us how the neural network works. It doesn't even work for a single neuron. So if you want to look at a 3-dimensional structure, you have to scan this focus in 3D, which takes up a lot of time and make it impossible to really follow the neuronal activity pattern inside the brain. So we use the concept of axially extended bezel focus, and by scanning this focus in 2 dimensions, we will get a projected view of the three-dimensional volume. The advantage is that now we convert the frame rate, which is the 30 frames per second, into an effective volume rate, as this example at 30 volumes per second. And we can use this approach and combine it with other two-photon microscopy module to achieve very high volume rate in vivo imaging. So here we are looking at a neuron whose cell body is at 600 micron and extending all the way to the surface of the mouse brain. With a conventional two-photon microscope, it requires to image more than 300 frames in order to cover this volume. But with this basal beam approach, we only need to scan this 100 micron long basal beam 6 times in order to cover the same volume. And with this, we were able to image in this volume, this very large volume, enclosing this whole neuron at about 3.4 volumes per second, fast enough for us to follow its calcium activity. And we can look at the individual spines within this volume, here about 145 spines, and looking at their activity pattern. So this would allow us to answer questions such as what is input-output relationship for individual neurons in the mouse brain. In terms of neural network, this method is also very powerful. Here we are looking at the inhibitory neuron population over multiple cortical regions in a mouse brain. The images I'm showing here are the conventionally acquired image using the three-dimensional scan or focus. You can see those individual spots are individual inhibitory neuron, and they are color-coded by the relative depths within this 1.5 millimeter by 3 millimeter by 600 micron thick volume. With the basal beam, we can scan this volume and their calcium activity simultaneously at about 1 volume per second. And we can then extract the calcium activity from this volume, and we acquired from almost 10,000 neurons simultaneously using this method. And at the same time of this measurement, we were also tracking the pupil diameter of the mouse with an enlarged pupil indicating that the mouse is very much aroused and awake. And we found that the spontaneous activity of those inhibitory neurons, some of them are positively correlated with the pupil diameter, therefore the arousal. Some of them were negatively correlated with arousal. And we can calculate the correlation coefficient between activity and the arousal level and color code each cells. And we can see a depth dependent as well as a cortical position dependent relationship here. And more recently, we also applied this method not to look at neurons, but look at the vasculature inside the brain. So here, using a conventional three-dimensional scan, we can cover this volume about 100 micron thick of blood vessels. But with the basal beam approach, we can cover the same volume, but at a much higher rate. And we still maintain the ability to see individual capillaries. So everything you can see with the conventional method, you can see it with the basal beam. Now, of course, the advantage here is that our volumetric rate is much, much higher. In this particular example, we were imaging the hemodynamics, the dilation and constriction of the blood vessels at 15 volumes per second. So in the last 5 minutes, I just want to discuss something that we are very excited about, where, when I was talking about this mind reading machine, we were able to achieve submicron spatial resolution with the help of adaptive optics. And two-photon and three-photon allow us to do this in opaque brains. And with the basal beam, we can image over very large volume, but we hadn't been able to achieve is this millisecond scale time resolution, which will require us to do two-photon fluorescence microscopy at a kilohertz frame rate. But more recently, we were able to do that in collaboration with Kevin Sha's lab at the University of Hong Kong, where they developed an ultra-fast line scanning method using the concept called free space angular chirp enhanced delay or FACET. So this is a very elegant method where this FACET module can be added to an existing two-photon microscope, where you basically have those pulse excitation laser. And in a conventional two-photon, we use this galvo scanning mirror to move this focus around inside the sample to form an image. The FACET module is a passive optical module made of a cylindrical lens and a pair of mirrors that are almost parallel except for a very small angle alpha. The cylindrical lens focus down the excitation beam and send it into this pair of mirrors. Now, if we look at this red beamlet, once it goes into the mirror, it will be reflected upon these two surfaces. And if the mirrors are perfectly parallel with each other, it will keep reflecting and go out from the other side. But because of this very small angle alpha, at each reflection, the incidence angle is reduced by the same amount. And eventually, it will become perpendicular to one of the mirror. At this point, it will retro-reflect and coming out this mirror pair and can be then sent to the microscope using a beam splitter. Now, the neat thing about this approach is that if you look at this gray beamlet, it enters into the mirror pair at a smaller incidence angle because of this cylindrical lens. And it will take fewer number of reflections for it to reach, to become perpendicular to one of the mirror and then back reflected. This back reflected beam now goes into the microscope at a different angle, which means that it will be focused at a different location. And because these laser pulses also propagate for a shorter distance, that means that this focus will also arrive at the sample plane earlier than the red beamlet. So another way to look at it is that this mirror pair forms multiple virtual images of this particular illumination point. And it is very similar in taking a picture of an object that is being reflected in between a pair of mirrors. Or if you are more artistically inclined, you may have heard of this Japanese artist and her infinity room, where a single lantern and multiple mirrors gives you this very mesmerizing array of these lantern images. Here, the lantern is one laser focal point, and the mirror pair generated an array of sub-pulses that are spatially separated and scanning the sample in one dimension, but also temporally denied so that we can separate the fluorescent signal excited by those individual foci. In other words, we do not need to scan the laser along the single dimension, and to form an image, we just need to use an orthogonal galvo to scan it in the orthogonal direction. And it's very easy to scan this even conventional galvo at 500 Hertz, which give us with bidirectional scan a 1000 frame per second frame rate. Using the genetically encoded voltage indicator developed by Michael Lin's lab, we were able to do kilohertz two-photon imaging, which allow us to see the spontaneous activity of a neuron inside the mouse brain. You can see the individual action potentials as well as the subthreshold responses. And here are just the example voltage traces from neurons at different depths. And you can see that we can clearly, again, see both the subthreshold and the single action potential. For this example neuron, we will observe about 100 action potentials, and we can look at the average for this and also 44 other neurons, rise and fall time of the voltage sensor response, which is very similar to what is known about this sensor. And each individual action potential corresponding to about 10% change in the fluorescence intensity. And we can look at also the sensory evoked activity, voltage activity. For example, there's a neuron we are looking in the primary visual cortex. You can see that if we present to the mouse visual stimulus that is not preferred by this neuron, then we do not see much voltage activity. But under the preferred orientation, we can see a lot more both subthreshold and single action potential generation. Now we can look at the tuning curve of the neuron using either the action potential or the subthreshold, and we can see that the action potential-based tuning curve is sharper than the subthreshold input, as expected and consistent with what is known using electrophysiological measurement. And here are just a few more examples of those neurons with both orientation or direction selectivity. And the very final slide, just as another example of this very fast temporal resolution, we can measure how long it takes from the time we turn on a visual stimulus to the time where we can detect the subthreshold input in the primary visual cortex. So here we are measuring this from 2 neurons at 345 micron depths. And it takes usually about 57 milliseconds for us to see this subthreshold response. And if we look at the action potentials, it's about similar time scale, about 60 milliseconds post the onset for us to see them. So right now we are trying to combine this facet method, which converts the line Galvo scan rate into frame rate, with the Bessel approach, which converts frame rate into volume rate. So hopefully, eventually, we will be able to look at the voltage activity at in a volume as well. So finally, just to sum up, here is my group, and the work won't be possible without their ingenuities and hard work. And funding agency, one disclosure, and thank you very much for your attention.